Hello and welcome to this video on one of the most common means of growing microbes, agar. Agar is a gel derived from seaweed. The gel is poured into petri dishes for microbe culturing. This is one of the most common ways of culturing microbes for diagnostics, quality control, propagation, and more. While it is the most common method, agar plates are not perfect by themselves. In fact, you can't grow much on the agar alone. It lacks the required nutrients and other materials for cell growth and propagation. In fact, this is why most plates are made using a nutrient solution with the agar added to it to make it solid. The contents of the agar can also be adjusted to make it possible for only certain bacteria or other microbes to grow. This means you can select for one type of bacteria, fungi, or other microbe and exclude all the others, or at least most of them. This is important for diagnostics particularly. You can grow pathogenic bacteria like Staph or Salmonella, and don't have to worry about certain other pathogenic bacteria growing in the place that you're trying to get this. And that means you can identify if one thing or another is causing a disease. By contrast to this, you can better support one type of microbe over another, rather than trying to exclude one. This lets you grow them to the exception of others because they have the most opportunity to develop. This can be particularly important when trying to culture novel species, the strains you have been developing, or those that have been obtained by opportunity. For example, strains obtained from old samples of beer, those that have been found in obscure and hard-to-access locations, and so on. With the need for different kinds of agar in mind, let's go through the major kinds that are used. First is very simply a blood agar. This quite literally contains blood, often from animals, although rarely not. Generally speaking, it's going to be something like a sheep or a cow. Most bacteria will grow on this, but you can identify certain phenomena happening from the bacteria that are growing on it, which can be particularly useful given the blood cells. They will behave in particular ways. It's also, because of its nature, a biohazard and therefore not recommended for most applications. This is why it is used for specific jobs. And then there is chocolate agar. A chocolate agar is a further refinement and development of blood agar. That's because it contains both factor 10 and factor 5. These are needed for hemophilus growth, and this is what it's used for primarily. That is, hemophilus and Neisseria growth. This is one of the major reasons why chocolate agar is used if these species are suspected versus just using regular blood agar. The extra materials involved in producing it adds to the cost, and therefore it's not used for all purposes, and only for those species that particularly need certain things present. Again, it's one of those selective applications where you can almost, by process of exclusion, define which species is causing the problem. And next we have McConkey agar, and this is useful in that it will separate out gram-positive bacteria from gram-negative bacteria. That's because the bacteria growing here can only grow if they're gram-negative. That is, they have a minimal peptidoglycan wall. Further, E. coli will turn into red colonies on this as there's a pH indicator present. McConkey agar also has a few other quirks to it, let's say. There are two versions available, one that has lactose in it and one that has no sugars whatsoever. Since E. coli will ferment sugar and turn it into an acid, you get that pH indicator change. However, if you use the sugar-free variety, you can add any sugar of your choice. This is useful if you're trying to figure out if something can ferment that particular sugar using this form of agar. For example, if you want to know if a sugar is digestible by a bacterium, for any purpose whatsoever, you can add that sugar and it's the only sugar source available. That means the bacteria can't metabolize it and use it. It's not going to change color, which is useful. Then there is Miller's LB agar. Now, LB is something we need to touch on now as it's rather important. LB is referencing to luria broth or lysogenous broth. We know it as luria broth, but that is more of a colloquial term rather than the actual name. The name itself derives from the original creator, and it's effectively one of the very first kinds of supporting media used for microbiology. The LB part of it, luria broth, is basically a nutrient-rich solution, generally made by taking animal products and a variety of other sources of nutrition and creating a almost soup or broth-like solution. Therefore, it can readily support most species. The only real changes between Miller's LB agar from others is that there are some variations in the exact proportions of certain nutrients and minerals within it, but otherwise it's largely the same. Neomycin agar builds on everything we've touched on so far, because now we're moving into selectivity based on introducing very specific material, in this case an antibiotic, hence its name, neomycin agar. As you would probably have already been able to pick up, the antibiotic of choice in this is neomycin. Neomycin is a relatively old antibiotic now, being discovered in 1949, so it is useful for identifying if a species has something like antibiotic resistance. But importantly, it is very effective at killing off both gram-positive and gram-negative bacteria. This means that you can identify something as bacterial by its presence. The other thing here is that if you are going to be using neomycin is particularly nasty for humans, and therefore relying on it as a first-line medication is not advised, and would only be used if the bacterium is resistant to other antibiotics and that it has proven to be effective on a plate like this, where the bacteria did not grow on it despite being exposed to the neomycin, whereas it is growing on other plates. Another application for the neomycin agar is to grow bacterium that are anaerobic. That is, species that don't rely on the availability of oxygen the same way that, well, humans would. In this context, they can get away by using other methods than the citric acid cycle. Another reason for it is that it also allows for Streptococcus species to grow more abundantly. Due to all of this, it can be quite a difficult agar to work with. Then there is non-nutrient agar. Now, non-nutrient agar is, for all intents and purposes, your absolute bottom baseline. That's because all it really does is provide you with a solid, or a semi-solid, surface on which to grow something. Now, this is useful if you're looking at trying to culture something that's not a bacterium. For example, viruses or fungi, even to an extent parasites or cells. Next we have nutrient agar, and nutrient agar is basically your level up from non-nutrient agar. That is, it gives the minimum required for a microbe to grow and survive, at least for a time. Unlike the other agar varieties we mentioned, it isn't ideal, and that's because although it provides a baseline amount of everything, some species may find it too rich and others find it deficient in what they need. And this means that it can be quite difficult to get some species to grow on this, and some just refuse to whatsoever. This is also a reason why nutrient agar is often used as a first test of kind. That is, if you're concerned about somebody being infected with something, or you're trying to grow a species you've found, you'll rely on nutrient agar first, and that's because you can hopefully grow it using the most universally available of options, without having to go to any great expense or difficulty in producing specialized agar plates. Sabouraud agar is another, but this is largely used for growing fungi rather than bacteria or viruses, and the reason it works well is that it has a low pH. That low pH is, in a manner of speaking, antiseptic, in that it will kill off any bacteria that you don't want. In some instances, it will contain an antibiotic. When this is the case, it will contain gentamicin. Gentamicin is, as mentioned, an antibiotic, but it's particularly good for gram-negative bacterium. Because of this, it's also not used as a first choice in most cases. When it is being used, it's often used as a means of identifying if something is gram-negative if mounting it on a slide and staining it doesn't produce an obvious result, although generally speaking, that's not a requirement. The next example we have is a Thayer Martin agar, and this is a chocolate agar, but rather than being used more generically, this is tailored to Neisseria gonorrhea, otherwise just called gonorrhea, and unsurprisingly, that's what it's testing for. The distinction here is that it's designed around the idea of the gonorrhea bacterium being a gram-negative species, and that it needs a certain support. This means that you can, at least in theory, very specifically narrow down if somebody has been infected with the STD. If you're looking for a more basic or fundamental media, you might try tryptic soy agar. This is another basic media used for culturing microorganisms or microbes. It's useful if you're trying to first start getting colonies going, that you can then transfer into something more substantial, let's say, or useful. One advantage of using this is that because it is a basic media and you're able to grow everything that you want and need, you can observe things like colony morphology, which is the shape, size, and color of the colonies. The round circles, although the exact size and shape will vary, and then you can move these on to other things, or if you need to grow up a sample in order to conduct biochemical testing, to conduct things like genetic testing, or if you need to store a sample for longer term usage. A tryptic soy agar may also not be used in agar itself, and the tryptic soy element may be used for a liquid culture. XLD agar, or xylose lysine deoxycholate agar, is used for poop samples, stool samples. The reason it's useful is that it has a number of indicators in it. Because it has indicators, you can see if there's a change. Now, the change is generally speaking going to be a shift into yellow, and this is because the lactose within it is fermented and you get production of acids, lactic acid, and that causes the change. The other reason it's used is that, to a certain extent, it will inhibit gram-positive bacteria, gram-positive bacteria having a thicker peptidoglycan wall, whereas gram-negative will not. In this case, we're thinking of something like E. coli, because you're also going to be encouraging the growth of gram-negative bacilli particularly. And because of this, it's also generally not used as frequently or as extensively. But if you get fecal samples, it's likely to be very commonly brought up. In terms of the preparation of these, you should find that you have a supply of petri dishes on hand, closed and put to a side until all the other preparation is finished. This is to avoid airborne contamination. Unfortunately, any open container will likely wind up with contamination unless you immediately put the agar within it. The alternative to this is to buy pre-poured agar plates. However, these tend to be considerably more expensive and tedious. Being able to produce your own on demand is generally more convenient in terms of affordability and practicalities, particularly for small labs. For larger diagnostic laboratories or those that do this regularly, they're likely to simply buy them in bulk and go through them quickly enough, it doesn't matter. Depending on the kind of agar plate you're trying to prepare, you're going to want to have your solution element, for example luria broth, available, and you're going to be generally making it in a 1 liter volume. From this, you're going to then add your appropriate additional ingredients. Agar is going to be the first, and depending on what we're talking about, this can be up to 20 or more grams of agar, although generally speaking it's going to be between 20 and 10 in each liter of your media of choice. To that in turn, you'll then add whatever your other ingredients are. For example, if you're making the luria broth, it would be 9.1 grams of tryptone, 4.6 grams of yeast extract, 4.6 grams of sodium chloride or table salt, and then your roughly 14 grams of agar. You'll then proceed to heat this up. Now, there is an important step in heating it up. You don't want to simply put it in a microwave and heat it up for 15 or 20 minutes until it boils. Rather, you want to heat it up for up to 5 minutes at a time, take it out, and then gently swirl the flask. This will ensure you get a consistent mixing of the contents of the flask, and that will ensure that you don't get a mass of agar at the bottom that's solidified. You continue to repeat the process of heating and stirring until the entirety of the contents is dissolved within the media of choice, whether that is just water with agar added, or water with agar and other ingredients, or a liquid media plus agar. You then pour this into your petri dishes. 1 liter of agar solution will make 50 large petri dishes at 100 millimeters each, or roughly 100 smaller petri dishes at 60 millimeters each. The pre-prepared agar that comes in a bottle is pretty much exactly the same process. The only difference is you don't need to add all the extra ingredients to finish the culture media. Rather, you just need to heat the bottle and be able to pour it after sterilizing the neck of the bottle entirely. This can then be poured directly into each of the petri dishes you're going to use. If you either have purchased the petri dishes or plates in bulk beforehand and therefore have them on hand, or you already have them in storage, you want to take them out of refrigeration where you'll be storing them and allow them to warm to room temperature before use. As far as storage goes, they should be kept in a refrigerated environment upside down. Now, this might sound completely contradictory. After all, upside down doesn't sound right. But remembering that anything on the lid of the container, the petri dish, will fall downwards using gravity. Therefore, having the lid on the bottom means that nothing can fall on the agar and contaminate it. The other reason is for condensation. That is, the cool environment of the fridge and the moisture in the air, even within the sealed petri dish, could still condensate and fall down onto the agar, and this could theoretically allow for the movement of bacterium, either contamination of the plate itself, or if you're trying to culture something on the plate, it could allow for movement of organisms between colonies, and therefore you get a contaminated result. Most petri dishes are going to be incubated somewhere around 30 degrees Celsius. This is about 90 degrees in freedom units. Each of the petri dishes is generally going to be sealed in something like a paraffin tape, or it may be isolated using other containers. When you go to culture the contents, it's going to be for somewhere between 2 and 3 days. Sometimes as little as 24 hours, depending on what's happening. Anything more than 72 hours, and you're likely to see results, but you won't be able to rely on those results because you won't know exactly what they are. The exceptions to this would be something like fungi, which grow very slowly in some cases, or certain parasitic organisms or viruses where you need that growth. Considering everything we've mentioned and noting that not only what you've been doing, how much the issue of biological hazards is present, and that some of the ingredients used for making the agar plate are, to be quite blunt, a biohazard, disposal should be on your mind. And one of the issues here is going to be, well, what you've been growing can be dangerous, so you want to destroy them, and this is why they're often disposed of using biomedical waste services. Another issue is that things like blood that are used to prepare them can be a danger, if not because of the nature of what they are, but that they can give rise to other organisms once they're disposed of as regular waste. So again, they're often disposed of as a biohazard with medical waste. And finally, you have things like the antibiotics that are within them. You rather obviously don't want to be putting random antibiotics out into the regular rubbish, and this is why most of the time everything goes into a biohazard bag. If it's not disposed of that way, then it nearly always all gets autoclaved. Now, autoclaves are the more effective option in terms of convenience. That is, you're not having to throw away material constantly, you're just disposing of what you have on hand. But of course, not everybody has an autoclave, and that's obviously a problem. In order to be able to use the petri dishes again, you need to clean them using a 20% bleach solution. And so this is rather strong, but by soaking it overnight, you achieve much of the same result as what an autoclave or biohazard disposal surface would do, but it takes 12 hours, whereas just about everything else can be as little as 2 hours or simply thrown into a biohazard bag. Hopefully this has given you a much better idea about the applications of agar, the different kinds of agar that are used, and some of the reasons why they're used and how they're used. Not just for the purposes of growing things randomly, but for diagnostics and to a certain extent for trying to make certain organisms grow better than others. This can be particularly useful if you're thinking about how to get something out of a very small sample you've obtained. For example, if you have yeast that you want to grow from something like a craft beer, this might be a means of developing it and continuing it on to something more applicable to your interests. Or you might just have a rather morbid curiosity as to what happens if you swab everything in a certain area and try to see what grows. Although, generally speaking, that's a terrible idea, mostly because once you do so, you probably won't want to touch a single surface ever again. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.